Thank you for joining us bright and early. First session of ARM's meeting on the Mesa, so appreciate you all being here. If you need a caffeine infusion, feel free to duck out and come back, no problem. We're excited to discuss today one of, I think, the major opportunities for the cell and gene therapy field. And I think ultimately everyone in this room is passionate about ensuring that patients can get access to these incredible therapies. That's why we do what we do. So we're really going to talk to some of the practicalities of how do we do that from here. And what I'm trying to get a movement started calling it CGT 2.0, like the next phase, because I think we've learned a huge amount from the leaders who have pioneered this field. But we know that we want to open up access and ensure that every patient who needs it can get access to these incredible therapies. So we're going to talk about what that means for the industry, how we ensure flexibility and scalability, which I feel, and I hope the panelists agree, is a requirement to get to that next phase. So I'm Jason Foster, I'm the CEO of Biotech, and I'm joined by a very illustrious panel with a huge amount of experience in trying to solve some of these challenges that span the full spectrum of development, from early stage R&D all the way through GMP manufacturing. So we're going to hear those perspectives now, but if we can, I'm going to ask my panelists just to introduce themselves quickly. Taby, would you start, please?
Sure. Thanks for having me, Jason. It's always a privilege. I'm Taby Ahsan. I'm VP of Cell and Gene Therapy Operations at City of Hope. City of Hope, for some of you who may not know, is a comprehensive cancer center, and so we really focus on that. But in the GMP space, we do a bit more than that, and I help spearhead the translation from our research arm into the clinic.
Ken, nice to meet you. I'm Ken Harris. I am the Chief Strategy and AI Officer at OmniaBio, and my main focus is on industrializing cell therapy in particular and whole cells. And I joined OmniaBio a little over a year ago, coming from Amazon, where I ran academic medicine for half a decade in the public health, where we were really thinking about what scale needs to look like in this space and how to move forward as quickly as we can to start unlocking patient access, which is quite difficult at the moment. So I'm looking forward to the discussion.
Andy?
Always a pleasure, Jason. Thank you for inviting me to this very interesting panel. My name's Andrew Snowden. I work at Johnson & Johnson. I'm responsible for aspects of CMC development for cell therapy, both autologous and other flavors. So really excited to join and have a conversation about what version 2.0 looks like versus version 1.
Thank you, sir. Tom?
Hi, I'm Tom Wilton. I'm the Senior Vice President of Innovation Ventures at the Children's Hospital of Philadelphia. Thank you to Jason for the invite to join the panel today. I've been at CHOP for just over 2 months, so I'm still learning the ropes, but clearly CHOP has been a pioneer in the development of cell and gene therapies, with the first approved CAR-T product, Kymriah, and the development of Luxturna. And through the health system today, we treat a significant number of patients with different types of gene therapies and cell therapies. So truly a bench-to-bedside capability, and looking forward to the discussion today.
Great, thank you all. I think we can all agree the science continues to move at pace. We're hearing and seeing incredible data coming out in new indications, so non-cancer indications, and expanding into other types of cancers that are currently untreatable in many cases, so glioblastoma, pancreatic, ovarian, some of these other very difficult to treat types of diseases. But I would argue that the infrastructure around these therapies just hasn't kept pace with the pace of the science. And too often developers are then forced into decisions about, well, do we try and maintain some flexibility before we get into the clinic? But how might that impact our ability to get to scale, impact our cost of goods, impact our throughput? All those things that we need to be concerned about if we want to ensure patients have access. So it's this kind of difficult trade-off that developers have to make between sometimes biology and automation, or flexibility and the desire to scale at some point. So the new standard we want to kind of put forward today doesn't force developers into that difficult decision. The idea is to provide flexibility and scalability at the same time. And we're going to talk about how we do that. And that really is a hallmark of what I would call CGT 2.0. So let me start, if I can, by asking the panel kind of a broad question to start. So we continue to make incredible progress. We continue to see, I think I was at a pre-discussion yesterday where the anticipation is we'll have we have 3 blockbusters in cell and gene therapy so far, one of which is the J&J Legend product, Carvykti. We anticipate having 10 more within the next 5 years. So the idea of being able to treat more patients and more indications is coming to a reality. But we know that difficulties remain. And so I'd like for you guys to help me in the beginning here to define flexibility and scalability in practice. What does it mean to be able to retain those two things? And Taby, I thought I might start with you because you're at the very beginning of the process, at the research and all the way through, obviously, clinical manufacturing. So you see that whole first transition. So tell me how you think about those two words as it relates to cell therapy manufacturing.
Yeah, so I think, for those of you who may not know, City of Hope really is positioned in that clinical trial space. And so we have a lot of clinical trials. And so I have 2 aims in trying to help this translation. One is get to the clinic as quickly as possible, right? So human data is the key for understanding your product and how well it will stand up to the test of time. So that's one element. The other element is that I want to remove hurdles from maturing that product to go to commercial. So how do we do that? We form platform with a little p, processes. These might be modular units. It might be A to Z as it works. So in our CAR T space, we have kind of an A to Z process that's pretty modular, and then we innovate on that. So we have a few of those platforms depending on how the cells are sorted, but that helps support 18 INDs for us. Just a couple of modular platform processes. So that's key to getting to the clinic quickly, right? Someone tries their product in this modular process that is already established, has already been qualified, and it's the home run. If it works, it's a home run. If it doesn't work, it's a pop fly. So that's always the concern, but at least you only spent a few weeks trying to vet that, or at least test it out initially before you invest all this time in process and analytical development. So that really helps on a couple of things on our CAR T space. It also helps on our viral vector space because those are really ones that favor this platform technology. A little bit different than our stem cell-based work, which is a bit more bespoke. But the key thing to those platform processes is making sure that they're not commercial processes, but they're compatible with commercial processes. Right. We don't want any elements there that would actually be a dead end for the product to mature to a commercial process. And so that's the key thing for us, is getting quick to the clinic and actually removing the obstacles of going commercial.
Makes perfect sense. Tom, at CHOP, how do you guys see a similar journey with your investigators and products?
Yeah, so I think similar in terms of how the cell-based therapies group approach early development of the process on a bench level and then trying to scale that up. Really our focus is taking those breakthroughs that we see every day at CHOP and taking them forward into first in human studies to prove that this works. And we have an incredible infrastructure for that, cell-based manufacturing, gene therapy manufacturing. To be honest with you, from a technical perspective, I'm probably less qualified than the other folks on this panel to discuss that. But from someone who's been in the biotech industry at pharma and in academia now, really when I think about flexibility and I look at the next 10 years, I see this amazing array of new cell products coming through. I mean, obviously we have the stem cell work that's been done, CAR T. My last company, Charisma Therapeutics, we were focusing on macrophages, a new cell type. Recently at CHOP, we've developed and delivered the first truly personalized gene editing therapies for a baby in the health system. So for me, you're going to see this incredible innovation coming through. And I have to say that the biggest challenge in all of my roles has always been manufacturing, whether it was in biotech, whether it was in pharma. We've seen incredible progress over the last 10, 15 years. commercial scale products now, but it still costs too much to develop these new therapies. The cost of goods is still too high, really, to see widespread adoption. With the potential in so many different areas for these products to have a big impact on patient care. So, I think flexibility moving forward is around defining systems that are interoperable and modular and that we can use for different product types, different delivery systems, different payloads, and academia working together with groups such as Ori and industry to take those from bench to bedside.
A lot of key points in there we'll hit during this panel for sure. Thank you. Ken, if I can come to you, some of those keywords that Tom just mentioned, I think are right in your wheelhouse around digitization and automation. As you think about some of those earlier development programs that you see, one of the words that or phrases that has become more commonplace in these forums is the idea of commercial viability. How do we create a process that can scale, that delivers the, not only the CQAs and the biological results, but also the business results in your TPP and your kind of what you're trying to develop. So how do you and your team and OmniaBio think about enabling those early companies to really future-proof their process early?
Yeah, I think it's starting with an early company, it's more helping them think about what an end state does look like. I mean, one of the things that we have done very successfully at Amazon is use what's called the working backwards process. Working backwards means you start with your end state, you define your end state, and all of the rest of the project falls out of that. And the end state, one of my criticisms in this industry, and I'm looking forward to the CGT 2.0, is that if you ask 90% of the people in this industry what their end state is, it's a clinically effective and safe product. That is one of the pillars, but that is not the only pillar. And we start our organizations with that in mind, and we fund them and we structure them with that in mind. And unfortunately, we are thinking much more like if you can go to the telephone system, we're still approaching that. That approach is, I'm going to go out and lay wires and switchboards and infrastructure for telecommunications versus I'm going to skip over that and go straight to a cellular-based system. Cell and gene therapy is not cellular-based, or is not cell phone based at the moment. It is still landline based, and we've got to get over that.
Yes.
So as we're trying to think about flexibility, we want to have infrastructure that's available for the 5-dose or 10-dose clinical trial, but at the same time be reminding the clinicians who are all eager and the scientists about their science, what does that really look like if you are successful and the dog catches the car? What does success look like for access? And access in cell and gene therapy is not going to CHOP or City of Hope. It is going to a community hospital and receiving that therapy every day to help you get better so you can continue to have your normal life. That's what access really looks like, and I can afford it. So we want to start to bridge that gap and do that through technology and data with people at an early stage. It's tough conversations, though, I will tell you.
Yeah.
No one's ready for it.
Andy is one of the only people in the room who have experience at both ends of the spectrum. So obviously J&J has a commercial CAR T product, but you're responsible for the next generation of products as well. Tell me how these factors that we're talking about right now play into your thinking as you think about developing the next generation of products for J&J.
Well, I think I would have to emphasize that you need to learn some of the lessons from the past. Very much what Ken was saying. You have to think about what your product before you go into preclinical development would look like if you're doing 100 starts a day in 3 different geographies and maybe 5 or 6 different manufacturing sites. How does it look like from the ground up for that to be actualized? Any single part of the process, from the most mundane to the most elegant, that is a pinch point, will just really be a problem. So you really have to focus with the end in mind on optimizing the workflows, using Kaizen principles in some ways, but really focusing on what would this look like in the manufacturing suite where the pinch point's going to be. Maybe it's media, maybe it's adding A, B, and C that takes 5 minutes in an R&D lab. That's probably 4 hours for 2-3 people per patient if you're doing it in GMP. You have to really think about true end-to-end systems and how do you drive down the complexity, down the costs, and how do you scale it? If it takes 18 months to tech transfer a process, or a year to 18 months to get commercial approval in certain geographies to go from 4 patient starts a day to 8 patient starts a day, you got a problem. You really need to figure out how to be able to move the dial and move the dial quickly. And version 1.0 has delivered breakthrough therapies. I mean, Carvykti is incredible. There are various other indications in lymphoma where you've seen really transformational results. But what we have to do is figure out where the lessons learned. And the big lesson is, if you have manual processes, batch records that are not streamlined and optimized, all these kind of things will pinch you, and it'll become a huge sink from the quality perspective, from the release perspective, from the actual scalability perspective of technicians, literally working under pretty arduous conditions in a full GMP smoke for 8-10 hours a day. You've got to figure out how to simplify. You have to think of the simple things. Calculation will take 30 minutes to check. Get rid of them. Everything that you can do to streamline it so that it can be done with less people, you really should do it. And you can improve consistency by doing that too. So it's a mutual win.
Yeah. Tom, at CHOP, how do we think about, or how do you guys think about embedding flexibility into the process? Because I think, part of your responsibility, as I understand it, is to take these products and find partners and, sort of market them to the world. But they need to have a certain set of boxes ticked, a certain set of criteria that would be, so it's possible for that. And I think we've learned the hard way over the last 3 or 4 years. If we don't take care of some of these things early in development, then those partnerships become more difficult and more challenging. So how are you guys thinking about this?
Yeah, so there's a number of aspects, as I mentioned. Looking at the breadth of different products that we work on at CHOP that we provide to our patients, certainly not all developed in-house. I think we have 10 different cell and gene therapy products that we now provide to patients in the health system. And some of those are developed internally at CHOP in the Center for Gene Therapy Manufacturing and the cell-based lab. And then a lot of them are developed off-site. So flexibility for us is really just being able to offer as many different, both approved products and products that are coming through in clinical, in clinical study as possible. And then the team working very closely with each of those different manufacturers, and whether they're pharma partners, biotech partners, CDMOs, to initially develop the process where we're developing that product in-house, or to work with them if it's already a commercial grade process on how we operate at scale. And most of that manufacturing will obviously be done off-site. So how the process from apheresis through to us receiving the final cell-based product, how all of that comes back to us. And it really, the thing that's amazing me coming into CHOP is just how many people that takes across the health system to make that work. I came from a 50-person biotech company, and CHOP has 30,000 employees, and a good chunk of them focused on advanced therapeutics. So you have to have the flexibility to work across different manufacturing platforms, different cell products, with different partners in different collaboration structures. And it, again, I come back to there's a technical aspect to it, and then there's a business aspect to it. There's a patient aspect to it, how we, ensure that, we can deliver these products to patients in the best setting for them. To totally agree all of the comments about that this needs to move from centers like City of Hope and CHOP into community hospitals, and I think that's really how these products ultimately will go mainstream. But I think over the last 10 years, it really has been more of the academic medical centers that have developed these processes initially.
Yeah. Taby, I think this is probably your day job. You're battling this sort of discussion, but how are you working with PIs and the institution itself at City of Hope to really ensure that this flexibility is built into that early research and that we are able to achieve those big objectives that you laid out for?
Yeah, I mean, it is definitely a mentality shift, right? In the early days, we might not be at 2.0, but we are in the latter half of 1.0. And in the early days, it was really a research endeavor. And many of the metrics that the PIs are trying to hit are still publications and grants. And at times, they would rather, instead of go to the phase 2 trial, go to another phase 1 in a different indication.
Right.
And that's because their role is different
Right.
than the role for those of us who are in the enabling space to get that product to mature. So we need, I think, those different components of groups within these AMCs in order to help have that breadth. You absolutely need all of that discovery, but you need a translation engine that is thinking not only about the next steps, but the next 20 steps.
Right.
And I think several of us have said it, and Ken very nicely talked about the end. And in that concept, what I talk about is not whether this is a scalable process, but I think about the end and I scale it down, right? That way I know that there is an end game for that product. But talking to these PIs who are so brilliant at coming up with these product concepts, we need to think about it not as a research program, but as a commercialization program for products in order to really get to that level of impact and access on the patients that we're all working towards.
Absolutely. And oftentimes when we talk about the idea of building scalable programs and scalable platforms, the idea of kind of modular automation comes into play. And Ken, I might ask you, if you don't mind, what's your view of where automation has a role, both in early development, but also in buying insurance against that future that you hope to be able to achieve? Because ultimately, we need to reduce variability, we need to shorten timelines. We're talking about tech transfer, we're talking about even manufacturing, full stop, and also ultimately delivering the biological result. All of those things are required to achieve that end goal, which is patient access and the big clinical impact. So how do you think about this, and how do you advise your clients when they come to Omnia, when they're thinking about this?
Yeah, I mean, I think if you think about the end result and learn from other industries and steal from other industries, automation exists and has existed for 20 years in other industries, and you can create a very complex product end to end with no humans involved in that. I don't think that's what we're ready for. I sort of always say it's always going to be a human in the loop, at least in my lifetime, in healthcare, just because we're classically trained that way, and until we all die off and the classically trained people go away, we probably won't get that out of the system. But we can introduce automation in certain areas, and I kind of introduce automation with concepts that are not very threatening. So talk about reproducibility. I had an industrial scale, FDA approved CAR T client talk to me about their product can't move from station to station and actually be shaken or vibrated because it changes the drug product. But humans are doing that. So they have humans walking across the floor from station A to station B, moving this thing. Now they're aware that they shouldn't be, but it happens, right? So you're like, well, let's take that out of the process. And what happens if we take that out of the process? Well, that would be a new submission because it's a change to the process. I don't know that that's true. We need to have those discussions with the regulators. But immediately you can see low-hanging fruit in manual processes where we can improve quality and probably improve cost right away that everyone can buy into. And if you start with that as one step, the second step is, PAT has been around half of my career in this space and really hasn't taken off in the industry. The reason it hasn't taken off is because we don't share our data. and maybe we don't even capture the data. It's even more concerning. But until we become a data-centric industry, and we all are scientists or clinician scientists, and it's amazing to me when I think about how we consider data, we consider it as proprietary and key to what we do, and we don't want to share it. Well, actually, as an industry, we need to start sharing that, because data will give us direction of where to think about tech and automation for low-hanging fruit. If we could start with some, industry cross-functional panels that are thinking about what data is out there. And it's not really going to come from the academic medical centers because they don't have enough data points. I mean, if you're doing 10 products, 10 trials, et cetera, for 10 patients, it's a very limited data set. We need to get into much larger data sets. And how are we going to do that? That's going to take biopharma, pharma, and biotech to lean into that. And I think that's our next real gap, is to think about data contribution and let the data lead us to what we should industrialize and automate and why.
I think that makes a lot of sense. Andy, I know you're thinking about these issues every day. How are you thinking about it, the kind of role of automation and, let's say, the next generation of technologies in your pipeline programs?
I'll spin off a little bit what Ken said here. Key thing is, how do you improve your reproducibility and rapid scalability? And it doesn't have to be the most complex automation pieces. It can be space saving, time saving, resource, FTE sync savings. These kind of things are huge. Also, I would say if you try and take even a healthy donor product and get 10 people to run through the process, or 10 teams, you'll see an extraordinary amount of variability because of the subtleties of the execution that can never be put into a batch record or training without them turning into a novel-sized document. So the key thing is that certain forms of automation should be able to cut through that, and is a great way of pushing this forward. And the key thing is, if you can develop this in a closed system, modular architecture, then you can have automation-compatible systems that aren't fully automated, kind of be delivered in a hybrid manual mode, where the key pieces of equipment are perhaps internally relatively automated with user prompts, with interoperability between the stations. And I'm very aware of if you change anything, it could be a comparability argument, but the key thing is, show it, prove it. I think that there's a lot of concern, sometimes warranted, about comparability, but until you actually go and look at it, you really don't know. And sometimes the most risk-averse approach is not the correct one. You've got to see whether or not a soft comparability works without presuming that it won't. And then you have to go from there to changing the processes over time if you really need to do an improvement that will increase your scalability, decrease your cost of goods, and perhaps decrease the number of out of specs so that you can treat more patients.
Is that really what good looks like? Are those the kind of critical success factors that we're looking at from a commercial perspective? Obviously, we need good biological product that's safe and effective. We take that as a given. But you need, let's say, higher throughput. We need to be able to deliver more doses. We need to be able to do so more consistently. So we're hitting our CQAs consistently. And we're doing so at a lower cost. Is that kind of we talked about reducing variability, which is embedded in that out-of-spec question. Is that what good looks like as we move into CGT 2.0, do you guys think?
Everything counts, yes. And it's strange to think about, but the most trivial pinch points when you start mapping the processes truly end-to-end and start thinking about scalability, those are the things that become your biggest hurdles. So if you can try and predict those and get them out of the way in your pre-IND kind of, or early clinical stage processes, then that gives you the runway that you don't need to think about comparability later, because the later in development you go, let alone When you get to a commercial product, the more difficult it is to prove that you're not changing something that could be critical. So everything counts.
Yeah. It seems to me like embedded in this conversation is a good understanding of the theory of constraints. I mean, I think, someone mentioned Kaizen principles. It's really mapping your process, understanding where the first bottleneck and then the next bottlenecks that arrive. I know, Ken, as you've been thinking about Omnia's next iteration into this area, you've thought deeply about, where robotics plays a role, where digitization takes a role. I mean, someone mentioned paper batch records over there, which always leaves me scratching my head of how do you release hundreds of products on paper?
8,000 pages batch records.
Exactly, with 2 people ticking boxes. That just gives me a headache thinking about. So how do you, Ken, as you think about, because I like the idea of we're not going to swallow the whole elephant, right? We need to take a bite and then take another bite and take another bite. So as you think about that journey from paper-based, manual, probably bag and flask type processes, how do you think about that journey, that, utilizing the thinking around theory of constraints? Because ultimately, I don't think, if I were to postulate, I don't think most people really understand where the bottlenecks are, the cost drivers in their process, and if they fix one thing, what's the next whack-a-mole to pop up? But I know you've thought deeply about this. And
yeah, in particular in CAR-T, which has kind of been my main focus over the last 6 or 7 years. But you could broadly apply this, at least in the autologous space, that there, as Andy mentioned, you start with the donor, and the donor has so much variability, and you're just out of the gates handicapped. So you can't go straight to automation and manufacturing and skip that point. It is the first gatekeeper. And how are we going to get there? We're going to get there with data and AI and understanding who our patient is and mapping our patient, and we can do that. My group, for example, has developed an AI program where we can, today, with 88% probability, we can tell you whether this autologous patient donor will actually meet manufacturing spec or not. Just by doing a phlebotomy draw and RNA sequencing on their T cells, we can draw that parallel. We'll put this into the clinic. My ideal goal is to bring that into the electronic medical record and actually empower the clinician with it, because we need to also get prescribing rates up. We're all here to drive volume. But prescribing rates and donor variability is sort of blocker 1. Blocker 2 is once it's in your system, that's really where your automation and batch records and digital infrastructure becomes really, really important. And you have to think about whether you're a decentralized or centralized based approach, and I think that's flexibility. I think the jury's out on what will win, and everybody has their preferences. It's probably not one or the other. It's probably going to be both. But putting a more automated system in City of Hope should be our goal as much as it should be putting a very large one in OmniaBio that can produce 20,000 doses. But that same small platform can scale.
yes.
And I think that's goal number 2. And then goal number 3 for us really is that last mile. And it's once I've produced the product, what all can go wrong? And lots of things go wrong, from sort of packaging the product to release, and release to logistics and it arriving at the patient. Believe it or not, it ends up in the wrong department, in the wrong floor, et cetera, et cetera. All those things, we can use technology today. If Amazon can get your package to you same day and go through 80-some thousand AI iterations to make sure the right way to get it to you, UPS, Postal Service, plane, et cetera, we can do that with cell therapy, and we can drive costs out by doing that. Part of the cost is in that supply chain. So I kind of break it down and start with those 3 big elephants and start chewing away at them on day 1, and that's what we're focused on.
Yeah, good point. It's not just one elephant. There's several of them in the room.
Yeah.
Taby, I know we've been fortunate to partner with you and your team and have you take a look at the Eero technology and think about how it might add value into some of the programs that you're developing. I'm curious how you're thinking about that that first step, what, as we're thinking about that iterative process of bringing automation in and trying to solve some of these problems, what are you thinking about and seeing as your team looks at, and you've got quite a lot of development happening in-house.
Yeah, I mean, we're trying a lot of different approaches. To your point, the jury's still out on centralized versus point of care manufacturing. City of Hope, we have, 5 national locations across the country. So right now we are centralized manufacturing because our GMP is only in one place. So how do we think about that, right? And I think to Andrew's point also, which is in order to increase access, you have to think about the COGS, but also the FTE time, the time in manufacturing space, the grade of clean room. Can we downgrade that to something that's a little bit less expensive to maintain? All of these things are elements of access and impact that are critical to think about. I'm hoping that we will have some sort of inflection point in a couple of years that will give us a little bit more direction on how we move forward. And that might be very much data-driven, right? If we can, not only, you talked about maybe the data isn't acquired or it isn't shared, we also have a problem that the data isn't looked at, right? I think a lot of the medical centers, we collect so much data and then we don't know what to do with it exactly, right? We need someone with your expertise to come in and help us really think about that from an AI perspective and how we leverage that to help direct that inflection point so that we are moving in a more effective way, more quickly as a field, not just as an institution or an organization. And I think that those are some of the key things that we have to think about as we're moving forward.
I mean, it really requires a new infrastructure. It's sort of, when Ken's talking about, well, we need a platform or a standard that's equally applicable at City of Hope at a clinical trial scale as it is to an OmniaBio at a commercial scale or a J&J. But, to Andy's point, 18 months or 24 months of tech transfer to try and get in between those two, time is money in drug development, and not only in, monetary value, but in patient lives. I mean, people are waiting for these therapies. So we need things that are interchangeable, that are comparable, you can scale from one node to many nodes or one large node. That flexibility has been missing up until now. And even tech transferring from one site that is under a pharma company's control to another site, one on the East Coast to one on the West Coast, or even from one clean room to the other in the same facility, takes a lot of time and effort. And those barriers are just, replete in the system and causing all kinds of challenges. So this idea of building ecosystems that can adapt to the needs of the next generation of science, this kind of idea of modularity, of flexibility, of digital connectivity, these are all embedded in that future. There's no way we could do it without those things. We know, people shaking bags back and forth like this in a GMP process is not adding value, right, shall we say? Creating more, creating more challenges than it's fixing. So, the way we're thinking about this is if we're able to reduce or maybe even eliminate process variability, then you're left with your patient variability. And then if you can start to work on that piece. But we don't actually know, I don't think. I mean, maybe Andy will correct me, but we don't really know what's driving the out of specs. The supposition is we've got different levels of health for the starting material in the patient cells, and that definitely causes variability. But when an operator takes a bag or a flask out of an incubator, goes under the hood, takes a sample, maybe gets distracted, turns an alarm off, there's some certain amount of time where we as human beings, no matter how expertly we are trained, are adding variability. We're adding process variability on top of that inherent patient-to-patient variability. And that at least is a place for us to start, if we can start to eliminate some of those variables.
I think, one of the aspects, though, which is you brought up, PAT's been around for a long time. We don't implement it nearly enough, if at all, right? Why is that? Well, are we not acquiring the data? Are we not analyzing the data? Do we not know how to analyze the data? Do we don't know how to implement it?
All of that.
All of that.
Yes, the answer is yes.
But layer on, also, regulatory agencies don't know how to regulate that process as effectively as a static manufacturing process. So there's a mentality shift. I said it before, but I think that there's a mentality shift in the field that needs to happen. Part of that is about data sharing, but how do we advance the field? And I think, right now it's it's a challenging time for cell and gene therapy, and this is when we need to come together and think about what benefits the field and not just our own individual organizations, and how we move forward in order to get to that inflection point where we're starting to see, we're getting out of the tow region and really starting to get to exponential growth.
I mean, one of the third pillars of our development platform is the digital piece. And, you know, we talk about data, sort of colloquially, we refer to data, but we also interchange that word with information. And actually, those two things are different. Data is different from information. We have a lot of data. We have very little information and almost no insight as to, like, what does it mean? And that, during our AMT application process with the FDA, we showed them some of the algorithms that we were building and where we thought there might be value. But that was almost ignored. We talk about biology, we talk about throughput, we talk about manufacturing. But there's so much we could potentially do, turning that data into information and insights.
And you have to be willing to do that. I mean, I'll give you a use case that we did. It was really, really simple, but the data was there and no one was utilizing it. So we were trying to understand why in knee replacement surgery post-op, we had such variability in patients coming out of the recovery room. It was huge variability. And recovery room time is cost, as well as recovery. And when we really started getting in the data, there was one surgeon that really stuck out in his anesthesiologist team as an outlier, that their patients came out of recovery really, really quickly and did very well post-op. And we went back and we looked at that, and guess what they do? They don't do any opioids pre-surgery. They only do high doses of acetaminophen for pre-op. That small little point was there in the data, but it had a huge impact on patient outcome, and it also had a huge outcome when we translated it over to cost of the surgery and of the recovery, and then socioeconomically getting the patient back to work quicker. It just ricochets through. And one little simple data point was there staring us in the face, but no one had even looked.
Right.
And we just need to look.
So thinking about this ecosystem that, you know, CGT 2.0 will require, Tom, maybe I can come to you and ask, you know, what is the role, from your perspective, of AMCs in respect to, also we have got CDMOs represented, we've got pharma represented, we've got academic medical centers represented here, and an enabling technology company. How do those different constituents play into the building of this new ecosystem?
Yeah,
and what's the role?
Absolutely. So obviously, a lot of the initial breakthroughs, the novel science happens in AMCs, the development of the next generation of cell and gene therapies, and been listening to all of the discussion as it goes on today, and it mirrors exactly what we've been talking about within CHOP, this interplay of, you know, really the teams trying to focus on doing the best they can for the patients today versus thinking about, okay, 5 years, 10 years down the line, and some of the new technology that's coming out of the incredible work done by different faculty members. From my perspective, I really see two areas. One is obviously as they develop those new technologies, starting to think at an earlier point about what is that path to commercialization, and that's very much what my team has been put in place to do. Things as simple as starting to develop a target product profile, which if you work in pharma is just obvious, that's what you do. If you work in academia, it's the Nature publication, it's the breakthrough. So trying to embed that at an early point, working with the cell and the gene product manufacturing teams as they start to do the process development work to keep that end in sight. And then something we've talked a lot about internally is really our approach to partnering. I think when we develop things initially internally, we have a good process now for reaching out with industry, with CDMOs, to work on scale of those. But oftentimes the new technologies, particularly platform manufacturing technologies, they won't be homegrown. So what's our approach to partnering with companies like Ori? And it can be challenging when you have a team which is literally walking the cells down the corridor to manufacture for a CAR T patient. You're trying to get hold of them to talk about, hey, we should be looking at this new manufacturing platform and let's do a evaluation agreement. They have different priorities. But I think there is an opportunity there for us, for other AMCs, to get better at data sharing, to totally agree on that front. Most of the data sits within organizations like ours. And B, then working with outside companies developing this technology to give a sandbox for them to be able to test that technology in a bench to bedside facility like ours, where you can rapidly iterate and you can enhance it. And we can get these new technologies to the point where we can actually start to use them in-house and they can have an impact. And if we're not taking some of our time to work on technologies outside of our four walls and to ensure that, particularly for CHOP as a pediatric group, pediatrics, unfortunately, because the markets tend to be smaller, are not always front and center when these new technologies are developed, so that they are suitable for the patients that we serve. And we'll only be able to achieve that by putting the time and effort into working with groups such as Ori as they develop new technology. So it's really, really important.
Andy, I think you and I were on a panel discussion, it might have been last year at some point, where we were posed the question about when's the right time? When's the right time to think about these issues? How early is too early? How late is too late? I think Daniel Palmacci was on that panel as well, as was Mark Bamforth. I'm curious if you don't mind sharing your thoughts on that, because I know you've got a lot of probably CGT biotech CEOs sitting in the room saying, I'm getting pressure from my investors to get first in man, I'm getting clinical data, I've only got so much money, this is all nice to haves. I need to get that thing that will unlock the next milestone. But I've got a thesis that the market's kind of voting with its feet if we get too far down the path without taking care of some of these issues.
So you need to get clinical proof of concept, obviously. But if you have a manufacturing process where you really haven't paid attention to CMC, where you actually have developed as a non-viable product, and increasingly people will look at it and go, there's going to be a big comparability argument that we'll need to make. There's going to be a lot of challenges scaling this, and just don't think it's worth the money. I mean, the reality is that you need clinical proof of concept, and you need something that should be able to be achievable with a reasonable cost of goods and a reasonable scalability to commercial, to the maximum patient population with as little pain associated as possible. Now, there's always work to be done, but if you're not paying attention to CMC and manufacturing early, people will look, people will notice, and it will be far, far more difficult for a clinical product to become a commercial product, especially as you're moving into these more challenging times for cell and gene therapies, where we really need to be able to scale as quickly, relatively painlessly, and achieve significant and continued cost of goods reductions. You really need to start with the end in mind, because ultimately the patients are waiting, and the challenge is that you need to make sure that your product is viable. And that means that I would argue that more than half of your product concept is tied to the manufacturing side rather than to the clinical efficacy alone.
Yeah, I think the investors are pulling back on that too. I mean, we say it's tough times in CGT, it obviously is, but also the investor intelligence and the way they make investments has changed over the last 5 years. And if you can just imagine, I just can't encourage you enough to think about your end state on day one, because when you're in front of an investor and you can talk about what your end state's going to look like. Yeah, we keep hearing that all they want is clinical data. They want clinical data. They want that milestone. They do, but they want to be successful. And they are getting very smart about what success looks like. And success is not just good phase 1 clinical data. Success is commercial success and viability. And if you are, as an entrepreneur, if you're having those discussions up front, the investor's going to be like, this is refreshing and this is different, and I like this, and I'm going to take a second look. We really have to reprogram ourselves. The FDA has done a great job of thinking that we all are tied to them and only them, but we are tied to payers, reimbursers, regulators in other spaces. And you need to be thinking holistically about who your ecosystem is in that end result. It's not just the FDA. And that needs to change for all of us.
And I think the FDA equally is concerned about this issue. They spend a lot of time working with sponsors to get INDs approved and products approved and go through the clinical trial process, and then only to see very few patients benefit from the therapies. And that it's one of the reasons why they have the platform designation, why they have the advanced manufacturing technology designation, is they want to see these products reach patients at scale. And so nice clinical data is, of course, a requirement to start. But to Tom's point, if you're building out, you need a TPP that has your commercial product profile laid out there such that, because ultimately if the product doesn't reach patients, what's the point?
Yeah.
What's the point of the 10 years and $1 billion of investment?
And we see exactly that. To your point, as an AMC, we're out-licensing a lot of technology. And so the clinical data or even the animal data will bring them to the table.
Right.
But more and more, their due diligence is super, hyper-focused on the
CMC, yeah.
And that is turning into deal breakers. And we see this actually over the last, I would say the last 3 years has really upped the ante on this. And the amount of time I spend in supporting due diligence for the institution is insane at this point because of exactly that reason.
Yeah.
They're getting smarter at how they invest. And to your point, they will walk away even if the clinical data is promising, the CMC doesn't support commercial viability.
Right.
Yeah, it's changed hugely. The first deal I did in the cell and gene therapy space about 12 years ago between AMC and pharma, it was all, we'll figure this out. We love the technology, and the manufacturing team will come in and they will figure out and do everything. And I put my biotech hat on from my last role. We had a new cell type. No one had made macrophages at scale before, and we had a number of very, very interested pharma companies. We got fairly far along the diligence process with them, and it was really the manufacturing groups there that said, hold on, don't do this deal, but we're going to have to do this, this, and this. It was using adenovirus. They were making lenti, not adenovirus. We've never made macrophages before, so it just puts the bar so much higher because they know what they're going to have to invest to be able to take that and scale it, and that's factored into decisions now, I think, in a way that it just wasn't 10, 12 years ago, so you have to be aware of.
I'd like to finish the discussion with a question for each of you. And Ken, I might start with you, and just to say, obviously, I'll put myself in this bucket. The difference between AI and machine learning, the distinction is a little bit lost on me. I'd like to say I know maybe a bit more than the average person walking down the street, but not much. How do we use these kind of, let's call them advanced analytics, either of those or others, to really improve cell therapy manufacturing? So I think there's a quality component, there's a cost component. These new analytical tools we found in our development can be very, even if it's just, to your point, presenting the operator or the people who are monitoring a process with information they didn't have before to make a decision on.
Right.
Even that kind of predictive opportunity is valuable. But how are you thinking about the application of these kind of new analytical tools?
Yeah, I would start, first of all, off the shelf. And anyone can use AI off the shelf. You don't need a background in it, et cetera. Andreessen Horowitz just published a very interesting article, I think on Thursday or Friday of last week, and they took all of their startup investment companies, and they wanted to know how they were using AI to accelerate their progress, and they mapped it out. And there were 50 different commercially available AI platforms that came out of this, and they listed them all in order of sort of spend of their own money through their investments. Number one and number two were large language models. And large language models can just help you with efficiency and things that takes me, 6, 8, 10 hours to do research on, I can literally do in 30 minutes now using a large language model. Yes, it might make mistakes, but I make mistakes too. So, when you go through that process, you want to start thinking about these tools are now available. Second is your data. You can have bad data, but I would start with using off-the-shelf tools to just push your data through it and have AI which is not necessarily machine learning, it's just analysis of data sets. Platforms like Palantir, Foundry are out there that can actually give you islands of isolation where your data has crossover and where it has uniqueness. And then as a trained person in the field, you can look at those islands of crossover and you can say, that island is very interesting to me. For example, looking at donors. Why is this one subpopulation of donors not the same as the other 90%? I want to look into that, right? So you can use those tools today. They're readily available. They're not really that expensive. And I would just start to get yourself comfortable with basic things like ChatGPT. Start asking the questions. And then as we expand on from large language models, now you want to go into like targeted AI. And that's where you need to start having skills, where you want to add the machine learning into it and say, I want to take this data set, and then I ultimately want to step out of the loop and let the artificial intelligence start to give me guidance. And I think that's where we'll get to PAT. It's ultimately, we will have machine learning in line once we've developed the sensors that will tell us, hey, I'm seeing these common warnings or themes in your batches that you're not picking up on, and you may want to dig into that, and the human can dive into it. So I think that's kind of like phase one. And we could advance this industry so quickly if we would just start there.
Start with phase one, yeah. I mean, we are applying those kinds of tools simply to tell you how many cells are in the bioreactor at any given time. We have inline, dissolved oxygen, pH, temperature, CO2, and we can tell you with reasonable certainty, plus or minus 15%, how many cells are in there at any given time in real time. And then if you want to take a sample and validate that, great. You can also make your model smarter on your process by feeding that data back in. But ultimately, I think where we'd like to get to is adaptive control, where actually the system then decides, based on that readout, do I do something differently? And of course, the human operator can be the one who says, push the button, yes, do, or no, don't do. But at least we have the opportunity then to translate that into insights that could hopefully lead to higher specs and better results ultimately with the...
I mean, I took our processes at OmniaBio, and with a very simple AI platform, I put all the human labor and all the process steps into this platform. I didn't build it. One of my colleagues built it for us, and he was able to identify 27% cost savings in labor, just in what we were doing, and saying, here, here, and here, we need to do it differently and compress it, or we need to automate it. 27% for us as a CDMO or for a pharmaceutical manufacturer, that is needle moving.
Massive, yeah. Yeah, for sure. And Taby, I know you and your team are thinking about this and things like, how much time does an operator spend on a unit operation or other things. Thinking about that kind of implementation phase that you're moving towards. What does that good look like in the next, let's say, 12 months for you at City of Hope in advancing some of this work?
Well, he's now set the bar at 27%.
28% it is.
Yeah, 28%. I think we're always looking at being early adopters of technology in order to try to figure out how we improve our processes. I think that, thinking about the ERO and thinking about other technologies and how we can implement them, remove the variability from the human side. With the autologous products, so we have a whole host of allogeneic products as well, but with the autologous products, that variability of the quality of the leukopak from the patient. And sometimes the product worked great for one indication. You move to another indication and you've just had a step change in the type of leukopak you're getting because that's a different patient population. So trying to understand, we are trying to incorporate these PATs so that we have smart manufacturing algorithms that can take variable inputs and lead to the same high quality output. That's the ideal, but that's always about trying to harmonize how we manufacture, as well as, because you need, in order to get that data to allow you to develop the PAT, you have to understand either that you're doing it consistently or what the variability is of what you're doing. If you are walking at 10 feet and it is shaking it a little bit, that is something that's part of the process. So really trying to dig in a way of what is controlled, what is uncontrolled. And then in this aspect of PAT, like you said, you can develop the algorithm and then the human can then, basically it acts as a flag, right? So the first step is have the data-driven flags, then the humans respond to it. Now the human response is actually part of the data set.
Right.
Right? And now you can start to iterate on that. And hopefully that's where we're going to get to.
A great final point to end on. Thank you guys very much for your contributions. And hopefully we've been able to focus on some of the key needle movers for all of you in the audience. We need to focus on our clinical data, of course. We need to focus on our commercial viability and think about how we incorporate flexible and scalable platforms into our development. So thank you very much to the panelists.
Thank you.